Not nearly as many people as we usually have. It's like there's a test going on or something. How many of you have already taken the test? Most of you. Okay. If you're here hoping for tests to help, you are in the wrong place. I cannot help with an open test, so good luck. Gonna pass the roll around. You guys know the drill. 8:30 on the front, 9:30 on the back. Just your name. So there's only like 12 of us, so it doesn't really matter, but. All right. Some good memes today. It's going to be a long 50 minutes, guys. All right. Biology fact for the day. The average person walks the equivalent of 5 times around the world in a lifetime. So that's pretty crazy, considering that really the only walking I do is from the ESLC to the engineering building, and that's about it. And I don't even walk that. I ride my bike. Otherwise, I don't make it in time. Study tip for the day is to reward yourself for sticking to a study schedule, or for maybe reaching the study goal that you had, or the grade that you wanted, the score that you wanted, right? This sounds like, a little bit childish, because we only really give like candy and treats and rewards to little kids, right? It doesn't have to be candy per se, but it can be, absolutely. Just give yourself some kind of reward for accomplishing it, right? Especially because in college, nobody really gives you treats or rewards anymore, right? And so you do well on a test, and the biggest reward out of that is that you got a good grade on the test. But you should also reward yourself with, I don't know, go out and get ice cream, or watch a movie that normally you don't let yourself have that amount of time. You say, I don't really have time to watch a movie. Pick a day and say, this is my reward for getting an A on that bio exam, or for hitting my study goals, every day this week. Whatever you got to do to just keep motivating yourself, right? Because nobody else is going to do it in college. Nobody else is going to be there to pat you on the back. Maybe your mom, but for most of you, she's not, in the same house anymore, so it's going to be a little more difficult. It's up to you, right? It makes a difference. Okay. Anybody have any questions on exam 2? Trick question. Some of you got excited. Those of you who haven't taken it were like, wait. All right, so we're going to talk about today's material. Those of you that actually came or end up watching the Penopta or staying on top of things, right? Because test 3 is in 3 weeks. So you got to stay on top of this stuff. And today's material seemed relatively basic, and it's something that most of us have heard in biology classes before. So what I always caution is don't get cocky, okay? Because by next lecture or 2 lectures from now, if you keep that attitude of going, I've heard this before in biology, or it's really not that complicated, big P, little p crossing, I make a 4-window square and yay, like it's not that hard. It gets exponentially harder very fast, okay? By next lecture, you guys will be going, wait. I don't know what those words are, I don't know what you're talking about, and he's not going to stop, and he's not going to slow down, so stay on top of it, okay? And again, I will reiterate, test 3 weeks from now. So, crunch time, right? And then test 4 is like a month later, and then the final is like 2 weeks later. So, there's not really like a downtime, although the other 80 people who are usually here seem to think that. All right, enough ragging on people who aren't here. Where's my pen? Uh-oh. Oh, it's like, who did that? This guy, Gregor Mendel, was a priest, basically, and he also was a gardener, and he realized that not all of his pea plants looked the same, and he started to notice a pattern, and he went, this isn't just random, right? I'm not just getting random different colors of pea plants just 'cause. there's some kind of order here. So he published his findings in 1866, right? And he said, I think there's something else going on here, and I think I can explain what it is. The genes in these flowers carry alleles, and he didn't use those words because at that time they weren't words. But he basically said the genes carry alleles, and when they mix together, they can give different outcomes, right? Nobody cared, and you still don't, but you should. Then in 1900, somebody else published it. I don't know whether or not they stole it. I'm fairly certain that they didn't steal it. They just also found it out on their own, published it, and they went, wow, this is genius. Look what you did. And Mendel was like, are you kidding me? He was also probably dead by then. So just know these dates. 1866 is when Mendel discovered his principles of genetics, and then in 1900 is when they were rediscovered and actually published. Okay? Just know them. Just be familiar with them. I think I remember a test question when I took this class, something along the lines of, Mendel published his findings in blank, and they were rediscovered and formally published in blank. And you just have to put 1866 and 1900. It's not super hard. It's not going to be like 1865 and 1901, 1866 and 1900, right? It's going to be like 1412 and like 2004, right? It won't be super hard, but so just loosely know those dates, okay? If you saw them, go, oh yeah, I know what that is. All right, so let's talk about the actual beef of Mendelian genetics. It starts with monohybrid cross. Okay, what's a monohybrid cross? One cross, but they're all one cross. One trait, right? We're looking at one trait. In this case, what are we looking at? Flower color, right? Purple or white flower color. Okay? We segregate our genes in the gametes, okay? They randomly fertilize, like we talked about, I'm going to say on Monday. Could have been Friday. And there are two kinds. There are dominant and recessive alleles, okay? Dominant being denoted by a capital letter, whether it's A, B, C, D, Z, whatever, and lowercase being recessive. It'll be the same letter for the same trait, right? So notice, purple's big P, white isn't like little w, right? P is for flower color, not purple. Big P is for purple, little p is for white, only because white is recessive to purple. You would think, well, wouldn't it make more sense to use a W instead of a P because it's white, and then we're less likely to get confused, and it'll be far clearer. But when you do genetics, it's not just with one trait, and so you can't just use a different letter for every color, because what if it's brown, but there's also blue, but there's also big, but there's also broad? Like, you're going to run into a lot of problems if you just use whichever letter works for you, okay? So it's usually the dominant allele, usually kind of logically lines up with whatever it is, so big P for purple, and then the recessive allele will just be the lowercase of that letter, regardless of color, height, size, whatever, right? Do you guys see where these letters are coming from? Nobody's freaking out about that? So let's actually step by step do a monohybrid cross. P generation means parent generation, right? So the first ones that we care about, the first ones that we start looking at, okay? This one has purple flowers. That's its appearance. Its genetic makeup is big P, little p, or sorry, big P, big P. The appearance of this one is white flowers with little p, little p. So what allele can this one give? How many options? 2? How many unique options? Better question. 1. If it gave this P, that doesn't work. This P or this P, it's going to be the same gene, right? How many unique options does that one have? 1. It's little p or little p, then they are the same thing, right? So in this case, we just care about the big P and the little p, right? They cross with each other, and we get, logically, big P, little p. But the flower's purple. Why?
Because it's the dominant trait.
Purple is dominant to white. So anytime a dominant or uppercase letter is present, that's what you get, right? Doesn't matter what other combination you have. If an uppercase letter, if a dominant allele is present, that's it. That's what you see. We'll learn some exceptions to that, but for right now, monohybrid crosses, that's the rule, right? We always end up breaking the rules. So now, how many unique options does just that one flower have? 2. It could give a big P or a little p, right? What are the statistical odds that it gives a big P? 75% chance that it ends up with a big P, but what are the statistical odds that it gives a big P? 50%. Random fertilization, or sorry, not independent assortment is a 50-50 thing. So this one or that one, 50-50 odds every time, right? Now, if it can give a big P and a little p, we've doubled the number of alleles we're crossing, right? Because now with the F1 generation, we cross it with another big P, little p, right? It's kind of missing here. That's how we go from here to here. So we take this big P and this little p, and we put them on the top of our Punnett square. And we also put them on the side, right? Because he's also contributing. It's not crossing with itself. These are sexually reproducing organisms. So it's crossing with another egg from a different plant with the same genotype, okay? So you just bring this P down and this P over, big P, big P. This P down and this P over, big P, little p. And you always just put the uppercase one first so that it's not like a whole hodgepodge of who knows what, right? Just always put the uppercase first. Okay, big P all the way down and little p right there, and then a little p all the way down and little p over there. That's how we ended up with a 3-to-1 ratio of purple to white. Somebody tell me how that happened. I basically just said it, but somebody else put it in your own words. What happened? Yeah, and the dominant trait, perfect. And one has the recessive. How'd that happen? It's an easier question than you may be thinking. Look at the Punnett square and just reason out how that happened. You mean these two?
Well, are you talking about the parents?
You're talking about up here?
Yeah, the F2 generation, both
Oh.
the F1 generation parents each had a big P and a little p.
Yeah, so they each had a big P and a little p to give, and so you end up giving a big P and a big P, or you give a big P and a little p, but since they both have a big P and a little p, they can also do that twice, and they can do that once. Okay, it's just the statistics of it, right? Now this isn't perfect. Genetics doesn't work on a 1-to-1 basis. There's no true 50-50 ratio or 3-to-1 ratio, right? And the dominant allele doesn't always show up more often than the recessive. My wife, her dad has blue eyes and her mom has brown eyes. Blue eyes is dominant to brown eyes. She has 4 siblings. Statistically, there should be 3 brown-eyed and 1 blue-eyed child, but it's actually opposite. She has 3 blue-eyed siblings and she has brown eyes, right? So it doesn't work perfectly, but more often than not, that's the kind of thing you see in a monohybrid cross, okay?
So like if we get a test question that has something to do with this, we're going to have to do like that process to figure it out?
You should, yes, be able to start from here, be given basically just essentially that information, and end up here and be able to get that. And we'll walk through how to do that. I'm not just going to say, all right, I'll give you 2 letters and now tell me the ratios. We'll actually, that's what I'm here for. If I didn't do it, I'd be a very crappy SI later. But yes, you should eventually know on your own how to go from there to there, given only 2 genotypes. Other questions on this slide? All right. Phenotype and genotype, 2 very important vocab words. Does anybody know what phenotype is?
It's the observable characteristic.
Yes, I like to call it the physical trait since they both start with a PH, right? It's the actual trait that we can observe, right? Tall, short, blue eyes, brown eyes, purple flowers, white flowers, spots, stripes, whatever physical trait an organism has, that's its phenotype, okay? Has nothing to do with letters, numbers, nothing. It's just, what does it look like? So then what would the genotype be?
Things you can't see.
Things you can't see, like,
Genetics.
yes, it's the genetic makeup, right? Genotype is the genes. So the genotype is that big P, or big P, little p, big P, little p, little p, little p, right? In real genetics, well, not that this isn't real genetics, but in more advanced genetics, we usually use more scholarly words and letters to signify certain genes. But for now, big P is its genotype. Or homozygous. What does that mean?
They got the same one.
Yeah, homo meaning same, so they have the same allele, right? Heterozygous then? Different. Hetero meaning different, so big P, little p. This one's also heterozygous. How's that possible? It's the same genotype, right? You might want to say, well, I mean, but why are they even denoted separately? It's because of the ratio. We end up with 2, right? So we have to point out that they're both there, but they are genetically identical as far as looking at the P's, right? And then homozygous again, but it's not the same homozygous as that top one. How's that possible? It's homozygous for little p, little p. So the fully correct term is homozygous recessive and homozygous dominant. Okay? So if I were to say a homozygous dominant flower is crossed with a homozygous recessive, you would have to know I'm talking about big P, little p, little p. And you could use whatever letters you wanted in that case because the letters just are there to show us what's going on, right? There aren't actually like P's and Q's and L's in our genetic code, right? There aren't even A's, T's, C's, and G's. That's just how we show it, right? So you should be able to recognize homozygous dominant and go, okay, I know that's 2 capital letters. I'm going to use R's because I like R's, right? And then I have also a homozygous recessive, so I'm going to use little r's, right? So even if you're not given the letters, know the vocab well enough to make the crosses, right? Trust me, he's not going to make it easy on you, right? He's not going to say a big R and a big R crossed with a little r and a little r. What do you get, right? Because first of all, you would just go, oh, it's a 3-to-1, because you could literally memorize that because that's the first cross you ever saw. But be able to go a little bit deeper than that, right? Yeah, questions on that? So phenotype, physical traits, genotype, genotype, we're done, is that, right? The actual genetic makeup, formerly referred to as homozygous and heterozygous. And there is no heterozygous dominant or recessive, because if it's heterozygous, it has to have a dominant trait, but it also has a recessive trait. So we just don't mess with that. We don't call it heterozygous, dominant, or recessive. We just call it heterozygous. Okay? Questions? I just stare blankly at the PowerPoint till I change the slide? Yeah, that's what I do too. I go to SI sessions too, guys. I sit in the back and I just... No, I don't. I participate actively. Segregation of the dominant and recessive alleles during gametic production coupled with random fertilization accounts for the 3-to-1 phenotypic ratio in the F2. What the frick does that mean? Somebody put that into understandable, I have a PhD in genetics words. Not taking shots at anyone, I'm just saying. Make that accessible.
It's basically just saying that the parent cell is going to separate, or sorry, the F1 generation is going to separate its two letters, and then from there, the sperm doesn't really care what egg it goes into. That's the randomization I was talking about. And then because of that mix, then you just get the 3-to-1 ratio of that.
Yeah. So it will just be an even split of dominant and recessive alleles to each side, right? What if they're both dominant? Because this is saying the segregation of dominant and recessive alleles. What if they're both dominant? What if it's big P, big P? It's almost a trick question.
Do you still segregate?
They still segregate, but it doesn't matter because they're the same gene, right? So I don't care whether it goes to the right or the left. I don't care which one it pairs with because it's the same gene, right? So what this is saying is we take a dominant and a recessive, and they are going to evenly pair with each other allele, right? They don't pick and choose. They don't say, I like this one better. There's no like chemical attraction that brings homozygotes together. Nothing. It is random. It is chance, right? But because there is that dominant allele, anytime there's a big P, you get purple. So that's how it ends up being 3-to-1, right? Shouldn't it be? Because if this is splitting in a 50-50 ratio, logically a lot of people used to think, well, they split in a 50-50 ratio, so it should be just a 50-50 ratio. That should be 1-to-1. But because of the dominance, right, that overpowers the little p, you always see purple if there's a big P, right? That's really what that's trying to say in way too many words. Questions? Not the most difficult concept ever, right? I promise you it's going to get more difficult because you're going to go from doing this to doing this. Right? Tell me how to do that.
So if it wasn't, if for example it was red and white and it makes into pink, for example, instead of going to the red color because of the dominant type of allele, would it then show like 3 or 2 to 1 to 1 or whichever one if it wasn't doing keeping the same color of the dominant gene?
What you're talking about is something called incomplete dominance. We get to that, I want to say, on Friday, and it's the idea that they kind of mix together, and we'll look at that later on, and it does change the ratio. So it depends on the trait, right? So in pea plants, purple and white are dominant and recessive, and there's no mixing. But in a lot of flowers, like roses, right? You've seen dozens of colors of roses, right? And you get a pink rose by crossing a white rose and a red rose, and you get sort of that mixing. That's incomplete dominance. But we'll talk about that and actually get into it on, I want to say, Friday. So hold on to that. Other questions? The other one is codominance. That's when they both show up. So like spots. If you were to cross like a, I think the example Mastering Bio will use is is a plain squash and a spotted squash, and then they cross, and it shows both traits at the same time. That's codominance. Again, something we'll talk about on Friday, but then when we talk about it on Friday, you'll go, that's what he was talking about, because it makes no sense right now, I know, but you'll see it. All right, so what is this principle of segregation that keeps coming up? Genes come in pairs that separate in the formation of gametes. Any gamete is equally likely to contain either member of the gene pair. Gametes unite at random during fertilization. Verbatim from class. Who has a little bit of a hard time understanding what that means right off the bat, just looking at it? You don't want to raise your hand, but all of you are looking at the slide like this. So I know. Okay, so it's saying genes come in pairs, right? Because we have how many chromosomes? 46, right? But they come in pairs. Chromosomes always come in pairs because they duplicate, right? So they come in pairs, but when we form our gametes, they separate, right? One to one, one to the other. It is an equal likelihood. Again, that's that 50-50 chance. Nobody's up there picking and choosing and saying this one looks best. 50-50 chance that it'll get either one. And then these gametes unite at random. That's not a perfect way to show that, but random fertilization, right? Any one of them is going to end up with another one. Big A, little a, little a, little a. Big A, big B, big C, little d, big E, small f. Like it, it's completely random. No matter how many genes you look at, that process is completely random. Okay? So just know 50-50 probability for segregation, and then completely random for fertilization, right? We can predict the ratio, the statistical likelihood. That's where that 3 to 1 comes from. But like I said, it's not perfect. This happens at random. So on paper, in a textbook, it's a 3 to 1 ratio. In real life, it's probably like a 2.78 to a point or 1.13, like some crazy ratio that's not perfect, but close, right? Did you have a question?
Yeah, so is this comparable to the second meiosis, like the second stage of meiosis?
Well, this is a meiotic process, if that's what you're asking, yeah. So this is because independent assortment and crossing over and random fertilization, those are all meiosis. So yeah, this is just, I guess, more of an explanation of how that part works. Someone else had a hand.
So the chromosomes, they're like attached together, the two chromatids or whatever. So does one of the chromatid have like one half of the gene and the other one have the other? Or am I thinking just
They each have one, yeah. Whether or not that's big A or big A, little a, that just depends on the organism. But yeah, so each one has one of the letters on its side. So then when they pull apart, the big A goes to one and the little a goes to another.
So haploids only have one of the...
Yeah, so like this would be a haploid cell. It just has the one gene. So that's like a sperm, right? And then the other one's an egg, and then when they come together, they put the two back together, right? Seems like a very long and arduous process just to stick them back together, but this is how we get genetic diversity. This is how we get stronger organisms, right? Survival of the fittest is genetic diversity, is meiosis, right? So that's why we want to do this painstaking process. Ooh. Other questions? Getting a little more complicated, and I don't think we actually looked at this in class, but I figured I'm almost out of material, so we might as well get a jump start. Are particular combinations inherited together? So does color go with color and size go with size, and eye color go with eye color, and height go with height? Nope. Right? It's always random. It's always independent assortment. So this side here is what would happen if dependent assortment were a thing. It's not, but let's look at it. it would always, always stick dominant with dominant and recessive with recessive. So dominant with dominant, recessive with recessive. Everything would be a 3-to-1 ratio, right? But we know that's not true because of independent assortment. So dominant can go with dominant, but dominant can also go with recessive. And recessive can go with dominant, and recessive can go with recessive. And then obviously the same copy down there. And we can get a 9-to-3 to 3-to-1 ratio, right? Does that Punnett square right there look like hell?
Yeah.
It got a whole lot more complicated as soon as we stuck one more trait in there, right? So let's practice. First of all, 2 to the n, excellent rule, is the number of possible combinations where n is the number of alleles. So how many alleles do I have here? 4, right? So 2 to the 4th is 16. How many traits do I have here? 16, right? So 2 to the n, number of alleles is n, always tells you how many combinations you'll get. Now, it doesn't guarantee that they won't be the same combination, right? That's just the number of combinations you can get. They may repeat themselves. So I found this handy-dandy Punnett square calculator because I used to tell people, yeah, so practice making your own Punnett squares at home, right? But who's going to tell you if you did it wrong? Nobody. Nobody's going to come and say, that's actually not how those were supposed to cross. You did that backwards, or you put the wrong ones with the wrong ones, right? Nobody knows. You don't know. So come in here instead and practice a little bit until you're comfortable making your own, and then check it, right? So it's really cool. You can show the frequencies if you want, and you can adjust how many traits. So that's a single trait, a monohybrid cross, right? That's a dihybrid cross. We're looking at 2 traits. You can go up to 3 traits. You go up to 4 traits. And this one stops at 5 traits. So if you ever thought genetics was simple, try and imagine the tens of thousands of traits that we can be crossing over, right? And then you go, making a human being should be impossible. And it really should. It makes no sense. Anyway, you can mess around, right? You can switch that to a big A, and to a little b, so you can get kind of a different practice there. You can say A is dominant, big A is dominant, big B is dominant, so that you can see, okay, this is a dominant trait, this is a dominant trait, that's a dominant trait, but it's dominant for A is red and dominant for B, or hang on, how am I saying this? Dominant for B is red and dominant for A is green. I assume those are the colors. And so mess around with this a little bit, right? So, because I haven't done a Punnett square in a while, we're going to practice one, and then we're going to check it against the calculator to make sure I did it right, okay? So, let's use different letters. So far we've used A, B, and P, right? Let's use D and what'll be good? You don't want to do, try not to use like O or X or ones that like you can't very easily tell. So like D's are pretty easy, right? What?
N?
Yeah, N is pretty easy, okay? So let's cross that one, right? I'll show you how to do one, and then I'm going to ask for a volunteer to come and do one, okay? Because we got way too much time. So, we do, it's true. Oh, hang on, I'm doing this wrong. I already set it up wrong. See? What did I tell you? We're going to do, let's do that. It already changed, right? It already got a lot worse. So let's do that with, no, that's not what I want to do. I'm trying to make this as difficult as possible Because I've always had the policy that if you show me the hardest way to do it, then I'm guaranteed to always at least be able to do it, right? So I'm trying to do like the hardest possible one. So, a couple of different ways to look at this. I pair up traits, right? So I see my alleles. I look at each one, right? That X is not an allele, that's saying that it's crossing, right? Then I take, and I guess I can show you over here a little bit easier. I take a big A, so I just start with the first one, and I pair it with a B, right? And then I get that. Then I take the first one again, and I pair it with a little b, and I get that. Then I take the second A and pair it with a big B, and then I pair it with a little b, right? So to do that over here, I would pair this one with that one, and then this one with that one, and that one. So that's going to look like big D, big N, big D, little n, big D, big N, big D, little n. Okay? Same thing over here. Big D, little n, big D, little n, little d, little n, little d, little n. So big D, little n, big D, little n, little d, little n, little d, little n. This is not my best work, but it functions, right? And then I just start pulling alleles in. Right? Now, someone else come try. Okay, come be brave. I'll give you an easier one. Let's do big D, little d, big N, little n, with a big D, little d, big N, little n. Somebody come cross that for me. Yes, brave soul. Here, I'll even.
Thanks. Appreciate it.
Make that a little bit faster for you. Go nuts.
Am I doing this right so far?
Yeah.
All right.
Oh, I thought you were gonna. So you've got the right ones in the right spots, except you've got to do the big D, little n, or
Oh.
big D, big N, and the little
Okay.
D, like little n, and then you could do like little d, big
Okay.
N. You see where I'm going with that?
Yeah. And then
So make sure the big D pairs with both of the n possibilities, and then the little d pairs with both of the n possibilities. And then just do the same thing for the next one.
all right. And then do I have that right?
You've done a big D. Is that a little n or a big N?
That's a big N.
So you're going to want that one to be a little n.
Okay. And then it's a little D.
Yeah.
All right.
I think you're going the right way. Yep. And then you fill in the rest mentally. You don't necessarily
All right.
for this purpose. But now you guys see the process, right? So you take the first one and you pair it with both possibilities, right? Then you take the second one and you pair it with both possibilities. Take the first one, both possibilities. Second one, both possibilities, right?
You don't pair the first and the second?
The big D, little d?
Yeah.
No. Otherwise, you'd end up with like, and that you would have, I don't know how to explain it, but no. You don't do it. Because the alleles have to segregate, right? So that principle of segregation that we looked at, they have to pull apart from each other. That's just kind of, I don't know, a law, I guess. We still have 10 minutes.
So once you have the Punnett square, do you just count up the ratio stuff?
Yes, then you, for now the answer is yes. We will learn a better way. But for now, because I don't want to spoil too much, you would just have to go through the painstaking process. Let's say D is for dark skin, and then little d is for light skin, N is for blue eyes, which makes no logical sense, and brown eyes, right? Okay? So you'd have to go through the painstaking process of saying, well, that's big D, little d, so that's dark. That's big N, little n, so that's blue. Then you would have to go on to the next one and say that's also dark and blue, and so on and so forth, and then count that up and find that ratio, right? We will learn a better way. Otherwise, one problem would take you 20 minutes, especially when you go on to 3 and 4 and 5 traits, right? Let's look back at this again. How much longer is that going to take? And then that, and then that. There's got to be a mathematical way to do that, right? That is not arithmetic. That is not count 1, 2, 3, right? We'll look at that. But for now, it's more just so that you understand the fundamental idea behind how we did that in the first place, so that when we show you how to do the math, you don't just go, yeah, numbers, that worked out. You actually know why we're doing it, but we'll get to it.
On this calculator, I see that they have the recessive allele before the dominant in their products section, so you have like a little a in front of the uppercase. Does that matter at all?
I think that's just a matter of programming for the computer, but at the same time, people do also prefer to do it this way, in the sense of they like to put A before B, because it throws them off to do big A, big B, big A, little b, and then big B, little a. So, and it's your Punnett square, so whatever makes sense to you. But most people will prefer to also keep it in alphabetical order. So when you start going, beyond one trait, then uppercase before lowercase, unless that breaks the alphabetical rule. Again, your Punnett square, do as you please. Use the letters you want. I wouldn't use numbers unless you want to use big 1, little 1, but that seems not helpful, and there's a reason we do it with letters, right? So again, literally just Google Punnett square calculator, go home, make yourself a Punnett square problem, right? Use whatever letters, try a tri-hybrid, right? Try a big D, big N, little n, big R with a big D, big N, little n, big R, big. Actually, let's shake it up. We haven't done any fully recessive yet. Okay? Consider that your first one. If you want to write that down and try that one as your first one. The same principle applies. The D has to pair up with both possibilities of N, but then also has to pair up with both possibilities of R. So you would get like big D, big N, big R, big D, let's see, big D, big N, little r, big D, little n, big R, big D, little n, little r, little d, big N, big R, little d, big N, little r, little d, little n, big R, little. Okay? It takes a lot more time, and again, that's why the mathematical calculation that you'll learn will make that a lot easier, but know how to do that, okay? And I don't just say that like we're running out of things to do in this session, so learn how to do that, right? That is something you will need to know how to do for exam 3. And if you go on in biology, you'll take a genetics course. right? You'll need to know how to do that. You'll go on to take microbiology, and you'll go on to use these things a lot. So I know today wasn't super complicated, blow your mind kind of stuff, but you have to know the basics before you can move on, right? Further questions? You guys want to screw around with a 5 hybrid cross? Look at that. But then it does simplify if you say that all the capital letters are dominant. You get a little bit more order to it. But it's fun stuff. It's what I do in my free time. I hope you don't believe me, because that would be very sad. Well, you guys want to just call it good 5 minutes early? Does anyone have further questions? What's that?
You want to do your pig wrestling?
Oh, see, I have a video of that, but it's disappointing to show it with so few people. We'll do it anyway. So I served an LDS mission in Ecuador, down in South America, and pretty much anything goes. It's a lawless country, and the audio connection. This is on the side of the road. Somebody I hope people are watching this. I should have prefaced this with I love it. If I didn't want to do medicine, I would have done it. I love it. I didn't really fight a pig, but he certainly thought I was fighting. I have another really funny one that I was going to show you guys. This one. They're all staring at me. This is on the side of a every single one of them. So I have a lot of videos. It would be considered unprofessional for me to show you them. No, they're not that bad. Maybe I'll show some more throughout the sessions. I actually have some pretty decent ones. Yeah, if you guys don't have any questions, get out of here.